Welcome to the Bioinformatics CRO podcast, where we talk to scientists, founders, and leaders at the intersection of computation and biology. I'm your host, Grant Belgard. I'm joined today by Dr. Nataraj Pagadala, founder, president, and CEO of LigronBio. LigronBio is a biotech company focused on molecular glue therapeutics, small molecules that co-opt the cell's own protein degradation machinery to go after proteins that have traditionally been considered undruggable. The company is applying computational chemistry, bioinformatics, and AI-driven platforms like its trimatrix analyzer to design these glues and target neurodegenerative diseases and other serious conditions where new therapies are badly needed. Nataraj has more than 2 decades of experience in computational drug discovery, spanning academia and industry from early work in biochemistry and bioinformatics through postdoctoral and research roles modeling protein structures and aggregates to senior positions in biotech and now founding his own company. Today, we'll talk about what he's working on now at LigronBio, how his career path led him into molecular glues and company building, and the advice he has for students, trainees, and scientists who are now thinking about careers in computational drug discovery or even starting their own companies. Nataraj, thanks for joining us. Great to have you on the show.
Thank you very much, Grant. Thanks a lot for giving me the great opportunity for the molecular glue audience and also for the targeted protein degradation companies. This is Nataraj Pagadala, founder and CEO of LigronBio. And LigronBio is incorporated in 2023, working on targeted protein degradation space, developing molecular glues for all undruggable targets in oncology side and also in neurodegenerative diseases, mainly focused on Alzheimer's. And later on, it will be extended to Parkinson's and also ALS therapeutics. So primarily, we are developing the platform called as the AI trimatrix analyzer platform to rationalize and discover molecular glues for the specific undruggable targets in Alzheimer's space. And also this is linked with the diagnostic kit, which is called as an LTAG assay. This particular LTAG assay will help in the functional studies of these molecular glues to take it for further for preclinical studies and also for clinical trials. So this is a powerful engine linked with generative AI that will help in discovery of these molecular glues within 36 months.
So for members of the audience who've never heard of molecular glues, what are they?
Molecular glues are the small molecules, which is all the medicinal chemistry properties are similar to traditional drug molecules, except that the difference between general traditional molecules and molecular glues are these molecules, they do the protein degradation compared to the traditional drug molecules where they inhibit them proteins in the biological system. So for the undruggable targets, basically there is no binding pockets where actually these undruggable targets help in the progression of the disease, even though there are the proteins which can be inhibited by the traditional drug molecules. So that is the reason why these molecular glues are designed especially for the undruggable targets for protein degradation.
When you explain your company's mission to someone with biology background, what do you emphasize first, the disease areas, the modality, or the technology platform?
So basically, our mission is basically to design the molecular glues for any of the disease-specific proteins, which is undruggable mainly. So, and at the same time, we are also, our mission is to do the targeted protein degradation for the diseases and also help in, reduction of the proteins in the biological system and also the disease progression. So our vision is very broad to develop a molecular glues for all the undruggable targets, and to save the future generations from Alzheimer's is our very big mission.
Are there any currently approved molecular glues?
Yes, yeah. So there is a couple of approved molecular glues. The two are, one is pomalidomide and also one is lenalidomide, which is in the market as a Revlimid. So for multiple myeloma. So and also it is a very big market for this particular molecular glue for multiple myeloma disease.
So what convinced you that there was space for a new company in this area?
So basically, if you see from the last 10 to 15 years, many companies are developing molecular glues in the targeted protein degradation, but unfortunately all these companies they are literally were not completely successful in developing molecular glues for any disease specific or also the target specific because of a lack of a serendipity. So this is the reason why LigronBio came into picture, we are developing because of serendipity reasons. The rationalizing the molecular glues and discovery of molecular glues is very difficult task. So we are developing right from the scratch. This is the primary reason why we are developing a trimatrix analyzer platform, where actually this particular platform rationalizes the molecular glues, and for a specific target, using a generative AI that will help in discovery one thing, and also at the same time this particular platform also find out all the off target interactions. That way we can eliminate all the serendipity problems within the biological system, to develop a molecular glue for a specific target without any off target interactions. That is the reason why LigronBio is a novel compared to all the existing platforms worldwide in terms of data integration with AI, and also high selectivity and specificity.
Neurodegeneration is notoriously difficult. What aspects of those diseases make them feel particularly well suited for molecular glue approach?
Basically, if you see in the biological system, in the neurodegenerative diseases like Alzheimer's, right? So that's what I'm saying, that there are many undruggable targets in the biological system that will help in the progression of the disease, not only in oncology side, but in also the neurological space, in the neurodegeneration. So these, as long as these undruggable targets exist in the biological system, it is very difficult for to inhibit the progression of Alzheimer's or Parkinson's and ALS. So this is where actually, unfortunately, the targeted protein degradation space is not introduced into this neurological space, and people are not successful as of now. So this is the where actually we need to develop these molecular glues, and eliminate these toxic proteins which are undruggable from the biological system. That way we can slow down the disease progression, and restore the memory function, and also reduce the cognitive decline. So this is where importance of molecular glue comes into picture with respect to neurodegenerative diseases.
How do you balance going deep on a few carefully chosen targets versus exploring widely across many possible targets with your platform?
So basically, this particular platform designs the molecular glues for any specific target. So even though there is no three-dimensional structures of the protein done by crystallography or by any other method, so this particular platform designs a molecular glue just by the amino acid. So basically, if you see the undruggable targets, then there is a motif called as a degron. So this degron is a 6 to 7 amino acids, or maximum 10 amino acids. So based on that, this particular platform designs a molecular glue based on the amino acid. So it is even the layman who doesn't know how to design the molecular glues, this particular platform gives an opportunity just by inputting the just an amino acid or a peptide sequence, it will develop a molecular glue. That's where this particular platform is completely different from all the existing platforms worldwide.
What kinds of collaborations or partnerships are most important for a company like yours at this stage?
So at this stage, particularly because the experiments of targeted protein degradation is different than the traditional way. So that is reason why we need a partnerships who are well versed with the targeted protein degradation space. So this is where actually we need the partners like a BMS who is working on targeted protein degradation, and also C4 Therapeutics or Chimera Therapeutics. These companies are developing are working on a protein degradation, but unfortunately they are not working especially on molecular glues, but they are working on other modality called as a PROTAC. But there are some companies who are working on especially on molecular glues, but they were not successful as of now. So we can help those kind of a companies. We can also partner with those companies to design the molecular glues with this particular platform, and also help them to for the targeted protein degradation with the molecular glues with our platform. That's where we can partner with those companies, and we can help those companies for developing a molecular glues.
When you think a few years ahead, what would success look like for LigronBio?
Earlier, a few years ahead, right? That time actually, to be honest, funding is much flexible compared to this particular time, where the funding is very bit hard. So because of not successful by many of the companies. So otherwise, by today, LigronBio might have developed a molecular glues for the Alzheimer's therapy, and by today we might have at least reached to the patients, clinical trials for Alzheimer's therapy, and also might reach the patients.
And is the vision to accomplish that through partnerships, or are you planning on sponsoring trials as Ligron?
Yeah, actually, we are also trying to do from our side our own clinical trials. At the same time, we are also looking for the big partners. Once we complete the initial phase of studies, once we file the IND, then we are also looking for the big partners, to step in and also do the clinical trials, as a joint collaboration with LigronBio.
What do you see as the main advantages and disadvantages of molecular glues compared to more traditional small molecule approaches?
The most important advantage of molecular glues is, because this is an event-driven mechanism, the effectivity and also the degradation therapy is more effective for any disease compared to the inhibition. That is the major difference between the molecular glues and also the traditional inhibitors, because the traditional inhibitors are occupancy-driven mechanism. So as long as you take the drug molecule, then the effect will be more on the disease state. But when in the molecular glue, even though the drug will be eliminated from the biological system, then still the effect will be more. So that is the reason why, if you see, the efficacy is also very high when compared to traditional molecules, and the effect will be 100 times more than the traditional drug molecule. So that is the reason why. And not only that, basically the molecular glues are treat undruggable targets, which is notoriously undruggable in the biological system and helps the disease progression. As long as these, as I said earlier, that these proteins are not eliminated from the biological system, the disease progression will still be there. That is the reason why we cannot stop oncology, we cannot cancer progression and also neurodegeneration. So there actually traditional methods cannot deal with those undruggable targets. Only molecular glues can help in that particular situation and help in the inhibition of disease progression.
What makes designing molecular glues hard scientifically or computationally?
Basically, as I said, the molecular glues, they influence the target protein based on a simple motif which is called as a degron. So degron is always as I said, maximum of 10 amino acids, right? So this is not a catalytic site. This is a catalytic site for traditional drug molecules is different than influencing the drug molecule based on this particular loop, which is a solvent exposed. So to formation of ternary complex is very, very difficult with respect to molecular glues. So this is where the difficulty comes in. One thing, because as I said, the degron is only 10 amino acids or maximum of 6 amino acids. So there will be serendipity of the molecular glues because most of the kinases contains this kind of a degron where 6 to 7 amino acids. That is the reason why there is a high chances of off-target interactions with the molecular glues. That's where we need to eliminate those molecular glues, and the AI trimatrix analyzer platform is the one that eliminates all these off-target interactions and gives them highly specific molecules for the that shows a target protein degradation.
How do you think about modeling ternary complexes and cooperativity when you're working with molecular glues?
So modeling, basically, as I said, we are training a very big database of ternary complexes right from the literature and also from our own in-house experimental studies. And we are also, mapping the proteome in the biological system for all the undruggable targets, so that will help us in, to see that using a generative AI, artificial intelligence, large language models, that will help us, to see that, how the molecular glue is especially seeing the off-target interactions. Once we eliminate that off-target interactions, it is easy for designing of a molecular glue for a specific target. So this is where actually that we are building the trimatrix analyzer platform, and also because most of the targets doesn't have a three-dimensional structure. This is where another advantage of this platform is that even though there is no three-dimensional structure, still we can develop a molecular glue for that particular target, just based on amino acid as an input. So this is where the advantage of this one, and also the difficulty that I said, in most of the companies, they don't have a three-dimensional structure, for most of the targets, unless there is no three-dimensional structure, there is no molecular glue. But a trimatrix analyzer platform can do this. And at the same time, most of the companies, to find out a ternary complex formation, they are using a diagnostic kits. Those diagnostic kits is based on the in the fluorescence. They only give indication about whether the ternary complex is formed or not. But when that is taken into experimental site, then it is not replicated. The diagnostic kit is not replicated. The results of the diagnostic kit is not replicated in the biological system in most of the cases. But we are developing a diagnostic kit which is called as an LTAG assay, which gives information about how the ternary complex is formed, which is like an alternative to X-ray crystallography. That's where we can clearly see that how the ternary complex is formed. So this is where the difficulty from all the big companies are facing as of now, and that's what we want to make it easier for all these companies with our trimatrix analyzer platform or also the diagnostic kit.
How do you decide which parts of the problem to treat as with more traditional physics-based structural biology approaches versus more data-driven AI ML approaches?
So basically, in the physics-based approaches, most of these approaches are for a traditional therapy for all the proteins which have a three-dimensional structure of the protein, right? On the catalytic side, there it is easy for the physics-based approaches, for designing of the drug molecules. But data-driven approaches, this where we don't have a proper three-dimensional structures of the protein. This is where actually the data-driven approaches comes into picture. Just like as I said, for all the undruggable targets where we need lots of data to develop one molecular glue for a specific target. This is where AI and also machine learning and artificial intelligence comes into picture compared to, even though basically artificial intelligence and machine learning is also useful for traditional therapy, but especially because that even though artificial intelligence and machine learning is not needed, still we can develop a drug molecule for the proteins which have three-dimensional structures of the protein and also the catalytic pockets. But without the data-driven approaches and without AI and ML, it is very, very difficult to design molecular glue for undruggable targets.
How important is experimental feedback for your models, and what does that loop look like in practice?
Basically, the experimental studies is very important because the important thing is very, very rare that we see the targeted protein degradation effectively by molecular glue in the beginning. So the experimental side is very, very important. Because there are many factors that we need to find out in the area of targeted protein degradation, especially with the molecular glues, because the product development is completely different. So it is easy to find out the targeted protein degradation with the PROTACs. But molecular glues is a small molecules and they influence the target protein through small motif. Sometimes, we don't know how the degradation is happening, whether the ternary complex is formed. This is very complex system through molecular glues. That is the reason why the experimental data, not only that, it's like, if you check, thousands of, hundreds of molecular glues, sometimes we end up with no molecular glue showing a targeted protein degradation. So that is where one experimental data and one targeted protein degradation will give a clue for many stages of a molecular glue development in the biological system.
Where do you see the biggest gaps right now in this space? If you could choose one particular type of data to just have a lot more of or better data of, what would that look like?
So basically, I see the main gap here is especially in the molecular glue is we don't have a ternary complexes. So that is where actually we cannot design a molecular glues ternary complexes, not only and also from X-ray crystallography, especially from the X-ray crystallography. Actually, how the ternary complex is formed, except 5 or 6 cases. Not only that, because when these undruggable targets, the ternary complexes form. It's a larger, it's a very big complex. It's very difficult sometimes to create a three-dimensional structures of the proteins through X-ray crystallography because of its complexity nature. So this is where actually the difficulty is coming from in the area of molecular glues. That's where we need to do some computational studies in the beginning with generate enormous amount of data what the ternary complexes, mapping of all the ternary complexes. That's where we get some clues to do the experimental studies. If it is replicated, then we can say that, this is what is happening from my computational studies, and this is also replicated in experimental studies. Then from that, generate more, molecular glues for other targets, more data driven through AI and ML.
So, to talk about your career, looking back, what were the big inflection points that shaped your career in computational drug discovery?
Basically, I did my PhD in computational chemistry in 2007. And after that, I did 4 years of postdoc in the University of Alberta and 1 year of postdoc in Belgium in Catholic University. So I have lots of my career, 25 years of experience. But, all my career, I worked on a traditional way, developing drug molecules for all the proteins, which has the binding pockets, have a very great traction record of computational drug discovery from the last 25 years, published for international publications. And also, I was also rated as one of the eminent scientists in computational chemistry by Carnegie Mellon University. So, but unfortunately, I never worked on this targeted protein degradation earlier before I started my career in bioethics. There my journey of targeted protein degradation has changed actually. Yeah, so from there, after going in-depth analysis, then I realized that, it's not a simple thing, I need to show to the world that, with all my experience that, how can we design the molecular glues? How can we not only molecular glues, how can, targeted protein degradation can be done easily. That is the reason why I started this particular career. That's where the inflection point has come in my career to show to the world that, how can we do this? Not only that, with the doing of this, how can we, reduce the progression of the Alzheimer's or Parkinson's and also ALS and also major this devastating diseases, with this technology, we can definitely protect the future generations, because we know that COVID-19 has pandemic has created havoc in entire world, right? Half of the world got wiped off. So that is the reason why I changed my career, that I want to do something to this, in the disease therapy, and I want to show something to this, how can we, stop the diseases, or also we can inhibit the disease progression and, protect the future generations for these devastating diseases.
What gave you the confidence to start your own company doing this?
So basically, my experience, from the last 25 years, as I said, I have a great track record of, computational drug discovery. And also because, as I said, I did 4-5 years of postdoc, PhD and publications, as from Carnegie Mellon University, I was also rated as an eminent scientist. So based on my career, my track record and my way of doing a drug discovery, so it's completely a little bit different, compared to other people in terms of thinking, in terms of implementation. That gave me confidence that, definitely my approach will help definitely for these diseases to for the disease progression, inhibit the disease progression. So that is the reason why with all my computational chemistry, because not only that, my other confidence is because I'm a biochemistry background. Mainly my background is biochemistry. With a genetics, with a PhD genetics department, and also I'm well versed with the molecular biology and all the biology aspects. So that's where actually I can easily connect my biochemistry experience with the computational chemistry experience, drug discovery experience, and also experience in biophysics. So with all these subjects, great expertise, it is easy for me to design the molecular glues, think about how the drug molecule works in the biological system. That's where I can easily connect. That's where my confidence has come that, I can achieve. Not only that, I don't need big laboratories to develop these drug molecules. I can sit at home and design the molecular glues on the computer with all my expertise. So that's where, I started this company because of all my expertise and also discovery of these drug molecules without having a laboratory spaces.
Have there been any particularly helpful pieces of advice from other founders or mentors that have changed the way you run the company?
Actually, because, there are very less people, who are working on molecular glues. So, and as of now, apart from the very big companies like a Neomorph and also C4 Therapeutics and also Chimera Therapeutics and BMS, apart from this, I personally feel that, I'm the only one who started as a startup with developing a molecular glues and developing a platform. Other than this, till now, I did not see any kind of other founder developing a molecular glues till today.
What's something about the founder CEO role that you didn't appreciate until you were actually doing it?
Yeah, actually, as I've basically, earlier when I was doing working in different companies, that time I was, my ideas was not taken into consideration. But as a CEO of the company, when I was developing this trimatrix analyzer platform, when I was developing this, designing the molecular glues, with a diagnostic kit, that's where actually people completely, seeing me as a different person in terms of because there are people who are well versed experience from the last 10 to 15 years of experience. Even though they have so much of experience, they were unable to figure out how the ternary complexes, how the targeted protein degradation is happening in the biological system, you know. But as a CEO of LigronBio, within a short period of time, when I was doing this, then people, are seeing me as a different, exceptional person, and then who can definitely deal these particular problems, help the community and help the society and also for future generations with Alzheimer's and also other devastating diseases.
From your perspective, what are the most underrated skills for computational scientists who want to work closely with wet lab teams?
With the wet lab teams, actually, we this is basically a different complex, biology. So I need, I want to work with the people who are well versed with, especially with the neuroscience one, especially with targeted protein degradation, who has experience in targeted protein degradation in terms of molecular glues. Without that, it's very difficult, to understand and do the experiments in the laboratory without having a knowledge about the molecular glues or targeted protein degradation. So I prefer the people from this particular background, if I want to work with. Yeah.
Where do you think molecular glues will realistically be in 10 years? A niche modality or something more mainstream?
Yeah, actually, molecular glues, as of now, molecular glues are in the high priority for different companies and also bigger companies like J&J. So because they are small molecules, as I said, they are brain penetrant, gut penetrant, and also membrane permeable. So molecular glues are the first priority as of now, and also till now 24 billions of money was deployed in molecular glue development by different companies and also by different VCs. So molecular glues are the highest priority in under the next 10 years. Molecular glues are going to occupy number one place compared to traditional drug molecules, because as I said, the effect of the molecular glue will be high, very high, 100 times more than a traditional drug molecule. So it is the first number one priority in the next 10 years. And also not only that, in the molecular glues are going to affect on the disease therapy, especially for the Alzheimer's. In the next 10 years, there is a high chances that molecular glue therapy will come into existence, for Alzheimer's, and also help the progression of, and also inhibit the progression of Alzheimer's. That way, it is a stepping stone for, reversing the Alzheimer's. If that happens in the next 10 years, trust me that, molecular glue therapy will also reverse the Parkinson's and also will reverse the ALS and also all the devastating diseases, even the cancer progression. We can reverse the cancer progression, and also we can inhibit the cancer progression, 30 to 40%. That increases lifespan of the patient and also the families who are affected with these devastating diseases.
Is there a misconception about molecular glues that you wish you could correct for everyone listening?
Actually, yes, basically people think that, molecular glues are very difficult to design. And also, molecular glues have a high serendipity and also off-target toxicity. This is what the people think about molecular glues. But, if you design properly right from the scratch, and also we can design a molecular glue with a high target, because last 10 years, this is what is happening with the molecular glues. Whatever the target is, basically they are designing, but ending up the same targets repeatedly every time, showing a degradation. So, because there is some problem in designing the molecular glues, that is the reason why we can design the molecular glues without off-target toxicity very easily if you do right from the scratch in a proper way. So this is the misconception that molecular glues cannot be designed so easily. That is a misconception there for the different companies all over the world.
Finally, if listeners remember just one thing from this conversation, what would you want it to be?
Yeah, LigronBio, we are unlocking the undruggable targets with Alzheimer's and the neurodegenerative diseases with the molecular glues. So this is where actually we are the pioneers in molecular glue discovery.
And how can listeners or potential investors connect with you to learn more?
So basically through email and also with my website, all the information is given in the website. And, please contact me if you want any kind of a collaboration, if you want any kind of an help in designing the molecular glues with a Trimatrix Analyzer platform. I'm here to help you in a very effective way. And also we can reduce the time of research and the cost of your research, and we can design the molecular glue for sure within less than 36 months. So all the details were given in the website. Please contact me, or else my email is npagadala@ligronbio.com and my cell number is 412-863-3812. Please contact with any of this medium. I'll be here to help you as much as I can. Thank you.
Nataraj, thank you for joining us.
The Bioinformatics CRO provides computational biology services to academics, research institutions, biotechs, and pharmaceutical companies on a flexible hourly basis. Visit our website, bioinformaticscro.com, to learn more.